Hi guys, I'm John, and this is going to be our video on the COVID-19 vaccines. This is a little bit of what we're going to be covering. And before we start, it's worth noting that there have been almost 450 million confirmed cases of COVID-19 as of March 8th, 2022. And in response to this, there's been almost 11 billion doses of the COVID-19 vaccines administered as of March 6th, 2022. Before we get started, we're going to look at a little background of vaccines in general. And to do this, I'm going to introduce you guys to Pascal.
Okay, so before actually talking about COVID-19, let's talk a little bit about vaccines and what makes them up. So, first of all, before vaccines existed, infectious pathogens were the leading cause of death throughout all of human history. Pathogens referring to microorganisms that cause diseases. These include things like tuberculosis, smallpox, malaria, and of course, the Black Death. Speaking of the Black Death, it's actually thought to be responsible for half of the deaths of all of Europe throughout the 14th century. So, before the advent of vaccines, the only way to become immune to a certain pathogen was to have been infected by that pathogen, survived, and then created lasting immunity for further infections. This is thanks to our splendid immune system, which is made up of our B cells and our T cells. These are our memory. Basically, the goal is to produce antibodies. As you can see on this graph, the most important thing is that on the primary infection, it takes a longer time to produce a given immune response. So looking at this graph, what's important to know is that upon first infection, it takes a longer time to produce a first immune response. However, after a second infection occurs from the same pathogen, you create a much more rapid, much stronger immune response. This is thanks to our immune system. So the logic behind actual vaccines is, if we can trick our body into thinking that it's already been infected by a pathogen, it will have already underwent that first stage of immunization, thus allowing us to either become immune or fairly resistant to further pathogens. Alright, so here are some of the current vaccines that we use for COVID-19. As you can see, right now there are a total of 8 leading vaccines that make up the majority of all of the doses administered. Of these vaccines, the most important ones are RNA vaccines and non-replicating viral vector vaccines. As for the mRNA vaccines, it's important to note that basically we take mRNA that is genetically engineered to encode for the spike proteins normally found on the surface of virions. We take this, we inject it into a patient, and then have the patient's host cells produce these spike proteins. In turn, what this causes is it causes our body to generate memory to this specific spike protein. Thus, upon further infection, we're already primed and we're ready to produce antibodies to fight off the infection. The viral vector vaccine is slightly different from the mRNA vaccine in that we actually take the genetic material from the virus, insert it into a non-pathogenic virus, and then inject this into our patients. Basically, this allows non-pathogenic viruses to infect the cells, thus causing them to produce the spike protein. However, this also leads to the generation of an immune response and the generation of antibodies. So this is slightly different from a protein subunit vaccine. In this case, Novavax is the most notorious and is currently approved in Canada. Basically, this is different because instead of taking the genetic material or making genetic material, we take the exact spike protein and then use that to vaccinate individuals. Thus, the protein itself stimulates an immune reaction, which can generate antibodies. But most importantly, the goal of all of these vaccines is simply to neutralize cells as they infect our body. Basically, these antibodies can coat the surface of these virions, thus causing them to not be able to interact with our host cells, thus preventing infection. Some of the common COVID vaccines that are being administered right now are Comirnaty's Pfizer, Moderna, Janssen, and Oxford AstraZeneca. Pfizer consists of 2 shots, 21 days apart, Moderna, 2 shots, 28 days apart, but both of them require a booster after 5 months. Janssen is slightly different that it was originally promoted by only being 1 shot, however current guidelines advise getting one of the other 2 mRNA booster vaccines after 2 months. This is slightly different from Oxford AstraZeneca, which is also a non-replicating viral vector vaccine, but it consists of 2 shots, 4 to 12 weeks apart. Additionally, there's some research going into a booster shot, which would account for a 3rd dose for the Oxford AstraZeneca vaccine. So now moving on to vaccine efficacy. It's important to note that efficacy refers to the specific ability of a vaccine to give a desired trait in a clinical trial. In this case, the desired trait is simply to reduce symptomatic infections of COVID-19. So Pfizer, being one of the mRNA vaccines, has a 95% efficacy, Moderna a 94% efficacy, also an mRNA vaccine. Janssen has a 67% efficacy. However, after adding the booster shot for Janssen, this goes up to 94% efficacy.
Next, we move on to the approval process. A typical approval process lasts around 6 or more years and consists of 6 phases, whereas an accelerated approval process lasts around a year to a year and a half, and yet still integrates those 6 phases in the typical approval process. The COVID-19 vaccines had to undergo an accelerated approval process because the FDA granted emergency use authorization to each vaccine after they were determined to be safe and effective. And although emergency use authorization was granted, it was not done before FDA scientists ensured that the benefits of the vaccine outweighed the risk. Despite the approval processes happening at a record pace, no scientific or safety evaluations were skipped over. A typical clinical trial has 6 phases, where phase 0 is a preclinical trial and a small number of people are given subtherapeutic doses and monitored for about a week. Phase 1 doses are increased in ascending dose trials to determine the most successful dose. Phase 2, large groups of people are given doses to determine efficacy of the drug and safety. Phase 3, large groups of people are given treatment to compare the efficacy of the new drug with the current one. Phase 4, there's continuous evaluation of the drug with a larger patient population over a longer period of time. In phase 5, patients are no longer monitored and the new drug is integrated into widespread clinical practice.
Okay. First we'll look at Novavax, which is a recombinant protein vaccine. They struggled to ramp up and maintain a safe manufacturing process after showing efficacy in phase 1 trials. They were one of the early players in the game, along with Pfizer, Moderna, and Johnson & Johnson. However, because of this, they never gained emergency use authorization. They've only recently been approved in some countries and are looking to be approved in the US soon. Next, we'll talk about Altimmune, which is a non-replicating viral vector vaccine, which is administered intranasally to the nasal mucosa. It showed poor immune response in phase 1 trials, and Altimmune abandoned development early on. Finally, Merck had 2 vaccines in development, which were both replicating viral vector vaccines, and they both showed poor immune responses in phase 1, and Merck had to abandon them. In general, these are a few difficulties that we're seeing in vaccine production. First of all, obtaining a sufficient immune response in clinical trials, which is normal in vaccine development. However, because of the magnitude of the COVID pandemic, there was more visibility on this process, and therefore it seemed like a bigger problem. There was also difficulty in establishing and maintaining a robust and safe manufacturing process, as was seen with the Novavax vaccine. We also saw that with the Johnson & Johnson vaccine, where a lot of doses needed to be thrown away due to a manufacturing error. And even after the vaccines are developed and shown to be effective, there's issues with storage and distribution of the vaccine, as was seen with the Pfizer and the Moderna vaccines, which both required very cold storage that made it difficult to distribute. And in the future, these vaccines might not be good enough because there might be need for a pan-coronavirus vaccine that would cover other coronaviruses.
This slide shows the risks associated with each vaccine. It is important to note that the expedited clinical trials do not allow one to know the full extent of long-term effects, so here are the known short-term effects. The mRNA vaccines are both associated with rare occurrences of myocarditis following the second injection, while the Johnson & Johnson poses a greater risk for developing rare but severe blood clots. Those with a history of severe allergic reactions to injectables or other vaccines should discuss the vaccination with their doctor, who can evaluate and assess their risk. The CDC noted that there are some situations when a Johnson & Johnson vaccine may still be used, if the recipient understands the risks and benefits. According to the CDC, anyone who has a severe allergy to any of the ingredients contained in the vaccine should not receive this vaccine. The mRNA molecules contain the genetic material that provide instructions for our body on how to make a viral protein. This triggers an immune response, causing our bodies to make the antibodies needed to protect us from getting infected, exposed to the actual virus. Johnson & Johnson uses the adenovirus 26 as a vector to deliver the DNA gene sequence, which produces the coronavirus spike protein. Lipids are required to deliver the mRNA to the cells. The remaining ingredients, acids, acid stabilizers, salt, and sugar, all work together to maintain the stability and shape of the vaccine. Thank you for watching our video, and if you'd like any more information, here are the sources.